Hello, my name is Laura Campano, and I will be presenting on this week's article, Spatial Transcriptomics Reveals Substantial Heterogeneity in Triple-Negative Breast Cancer with Potential Clinical Implications. This was published in Nature Communications. A little background on this article and what they were studying. They were studying triple-negative breast cancer. The triple-negative comes from the fact that it does not have receptors for estrogen, progesterone, and HER2 protein, which decreases the amount of therapies that can be used, such as hormone therapies. It is a heterogeneous disease, so there'll be molecular subtypes and they respond differently, and they also vary in aggressiveness, so that makes it more difficult to treat. So a quick summary on what was done in this research. 96 triple-negative breast cancer samples were used to analyze their spatial transcriptomics using histology slides as well as the bulk RNA sequencing. Morphological features were then annotated and then grouped into clusters using bioinformatics, and that helped the researchers visualize what features were in the environment of the tumor cells. The amount and spatial organization of tumor-infiltrating lymphocytes as well as the lymphoid aggregates have been useful in facilitating more effective chemotherapy and immunotherapy responses for a disease, so they can track progress on these diseases. So the goals of this research was mainly clinical application and how to treat cancers and what are more effective ways of treating cancer. So they gathered more context of the transcriptome of triple-negative breast cancer. They studied the cell's environment using the spatial transcriptomics. They studied how each subclone varied genetically, how the space in the environment of the genes and how they impact the tumors. They gathered more information about how the clinical treatments respond to the tumor cell and ways to increase the treatment's effectiveness. They also looked at the composition or organization of the tumor and stroma, because that can tell you how deep the cancer is, or if there's immunity activity, or seeing like how much of the cancer is progressing or getting better. So these triple-negative breast cancer molecular subtypes include basal-like, which has a moderate recovery rate, has a deficiency in its DNA repair gene, so which causes the genes to be unstable, and increases the amount of TP53 mutations. Immunomodularity has shown an increase in immunity signatures. Luminal androgen receptor expresses androgen receptors and frequently mutates in the PIK3CA gene, the AKT1 gene, and CDH1 gene. And mesenchymal and mesenchymal stem-like molecular subtypes are more aggressive because they can use angiogenesis to grow more, and they also see stroma signatures. So here we have a visual of these subclones. Immunomodularity, an increase in immune signatures. The basal-like, increase in genetic instability, DNA repair genes, and TP53 mutations. They're able to see a high amount of immune activity in these ones. And then the more aggressive tumors, such as the luminal androgen receptor, which has an increase in androgen receptor expression in the PIK3CA mutations, mesenchymal, increase in angiogenesis and increase in stroma. The mesenchymal stem-like shows a decrease in mutational load, so the stem-like is easier to treat as compared to the mesenchymal. A popular methodology that transcriptomic research generally uses is RNA sequencing. The limitation on that is you cannot visualize what's happening in the cell, and that is important in terms of disease progression and treatment. Spatial transcriptomics allows for geographic intratumoral heterogeneity, which helps with assessing how the immune system is interacting with the tumor. So looking at lymphoid aggregates, if there's an increase or decrease in that. Epistatic interactions between clones of tumors. How various genes impact other genes in tumors. You saw that some subtype clones had a lot more mutations in different genes, while others only had it in one specific region. That's an important thing to study from a heterogeneous disease. And you get more context on the tumor microenvironment with this type of method of spatial transcriptomics. So you can see where the immune cells are located and the interactions between other cells. I added this visual. It is not from the article I am presenting on, but I just thought it would be a good visual to see how complex this type of cancer can be and how it can look under a histoslide. So the methods for this article included sample collection from cancer patients and turning those samples into histoslides. Then those histoslides were used to mark morphological features, and then those features were counted up in software. 96 triple negative breast cancer samples were turned into histology slides, capturing gene expression across multiple tissue regions of each sample. Histological annotation in double CD3 and CD20 immunostaining was used for tumor structure identification and tertiary lymphoid structures, which that shows immune activity. Transcriptomic data was collected to identify spatial morphology and immune activity. Spatial deconvolution and clustering revealed the types of regions of the tumor and stroma, and that led to the signature of a good treatment response of the 30-gene TLS signature. Spatial transcriptomics, this is what it looks like. The two visuals I put on the presentation is color coordinated, each pixel to show how much of the region covers what type of immune activity, tumor patches, and stuff like that. We can see where genes are expressed using these spatial coordinates with intact tissue sections. You can see what's stroma versus tissue, and identifying immune selectivity. And here are the results I collected. There's the HEE stain, and they also showed what it looked like as it was annotated. They have the categories, so it's an easy visual to see how much of each activity is occurring in every cell. So we see in the first one, you see a lot of tumor cell activities, and then the next one you see more stroma and lymphocytes. So that could be indicative of, progressive or a weaker cancer as compared to the first one. Those pixels were counted up and put into a bar graph, so you can see how much of each type of morphological features were present in these histoslides. We also counted up the samples that had so we have, all of them had tumor cells, but only a very small amount had nerves present, so that the ones that have nerves can be indicative of the subclone that utilizes angiogenesis. This visual that the article provided brought more context on what regions were expressed in each subclone type. So how can spatial transcriptomics be applied? It can be applied in more personalized cancer treatments, because thanks to seeing the microenvironment, we can identify targets and barriers in cancer cells, how far cancer activity is occurring versus just the stroma. Routine analyses can be used to see the progress of a tumor cell versus immune system activity. The lymphoid aggregates are a good indicator of if there is if the body is fighting off the cancer. Modeling epistatic interactions with the progress collected, and further research on the triple negative blood cancer molecular subtypes. For references, the Stewart citation that is for the image I showed earlier of the various histology slides of triple negative breast cancer. And the second one is the article that we have read.